The challenge of stem cell research, many hurdles remain. They asked me if I can make 2 presentations here, so I came just like that recently from a meeting in Boston, in Summit Stem Cell in Boston, when I gave this conference about the stem cell, okay? So, I'm from the University of Texas Health Science Center at San Antonio in a diabetes division. So, I won't like to say that science is provisional, always open to revision, in light of new evidence. It's an anti-authority system. Anybody can contribute. Anybody can be wrong. He seeks actively to test his proposition, and he's comfortable with uncertainty. And it's like what you said, American Diabetes Association don't have the certainty by himself, okay? That's science, okay, by definition. So, encapsulation and stem cell are not anymore new field of research, okay? You have to know that the differentiation of new islet from stem cell or precursor cell had been active since the late 19th century, okay? It's not a new phenomenon, okay? And on 1911, a study on the pancreas of a guinea pig demonstrate that, okay? So it's not a new phenomenon. It's known for a long, long time, okay? Sometimes I said we love to say that we make something new, but it's not necessarily new. It's like technology wireless is a radio wave, okay, discovered by Hertz, okay? But we call that wireless. It looks new by definition. So, molecular biology, okay, molecular biology discipline originated from the 1930, 1940, became institutionalized in the 1950 and 60 with the discovery of the DNA, okay? And molecular biology will produce the marker to identify the stem cell, follow the lineage of cell. Molecular biology brings what I call the reductionist thinking into medicine, and that is prone to fallacy. For more than like 2 decades now, we definitively lost contact with biology. And we're thinking too much reductionist, okay? And that brings us to fallacy on this area. And if you take an example, the reductionist thinking of gene expression to follow the cellular lineage. If you check the stem cell area, you will see all this lineage, okay? But in reality, what you have here is a fragmentation of knowledge, okay? And it's a really fragmentation of knowledge. It's not because the expression of the gene X is present in adult beta cell or in a development pancreas that is an evidence that is directly affect the differentiation of the beta cell, okay? It's a fallacy by definition. And the problem with the Krelux lineage targeting all the cell in embryonic cell, so we love this transgenic animal, okay, but also all the cell have the marker at the embryonic level, so it's impossible to differentiate between who is the cell coming from. And the conditional Krelux strategy have the problem that the promoter specificity, okay, that is expressed mosaically, okay, and also the Krelux toxic, the protein is toxic by himself, okay. And the knockout gene suggests but does not confirm the implication in a gene in development. It's an error also in the modern term when we're using this kind of technology. Just to give you a schematic view of the complex of gene expression regulation in the nucleus, okay? So here, when you check how the gene work, okay, an example here. So if you take this gene, for example, reductionary linear thinking doesn't apply here, okay? This gene will have an impact on this one, and this one. Another gene will have an impact on this one expression. Sometimes you will have autoregulation when the gene is expressed, will have an impact on the other gene and vice versa. So in reality, when you use knockout gene, and when you use all this Krelux system, you simply messed up what's happening in vivo. You simply messed up with that and they give you absolutely nothing what's happening in vivo. You have a fallacy error to thinking that your approach is linear, and it's not linear. So, the non-linearity of the biological, So the non-linearity in a biological system makes it difficult to follow cell lineage, okay? You have to understand that 90% of the primary transcript never translocate to the cytoplasm. They simply degrade and ritualized by the nucleus again, okay? Only 10% take off outside the nucleosome, okay? And after that, you have to understand that the transcriptional level never correlate with the RNA level. It's an error to think that that part are linear. mRNA can fluctuate like that, and your transcription can fluctuate the other side. You have also that the mRNA level never correlate also with the protein level. It's an error that is made systematically here. And the protein level never necessarily correlate with the protein activity level, okay? So still today, an emerging challenge for life science research is to identify the field of genetic and physiology. And it still stays a challenge today, a huge challenge. And everybody are their own recipe to form beta cell from embryonic stem cell, okay? You can have in the literature more than 100 different protocol recipe that produce insulin-producing cell. From monocyte, hematopoietic stem cell, mesenteric stroma cell have been published. But all these different recipes to produce insulin-producing cell pinpoint the curiosity. The curiosity of all these experiments that poorly mimic the natural pathway in utero of the development of the pancreatic beta cell, okay? So in reality, we create simply chimeric cell like Drosophila experiment. Drosophila was the first fly utilized for the development. We have so much experience that was spectacular in the note if you are in the 1980 when you put the head, arm on the top of the head of the fly, you change all the thing like that. But in reality, it was curiosity. It doesn't happen in the nature, naturally in the nature, okay? We do the same thing in our laboratory. We create chimeric, but in reality, that is not necessarily reproduce what's happening naturally in our context, okay? So the problem of extrapolation from cell lineage technique, okay? The signal that promote islet development in utero are poorly understood still today, okay? And although the current protocol allow formation of beta cell like critical signal that promote the final step of differentiation are still missing. And our knowledge on human pancreatic development is derived largely from animal model, mainly mice, with the assumption that the same mechanism molecular pathway is conserved in human, okay? Working at the gene level, an example, at promoter level, my cell, okay, I can tell you that the insulin promoter, an example, band some transcriptional factors that are the same in human, but they are not disposed in the same place. And more than that, you can find also transcriptional factor in the mouse model that bind the promoter that don't bind the human promoter. So you have a difference here, okay, by definition. So further optimization of the existing protocol to generate beta cell without using oncogenic factor is also needed, okay. At this moment, an example, you have the problem what we call the tetramer formation linked to failure treatment with type 1 diabetic using embryonic stem cell derived from insulin-producing cell, okay. After transplantation of insulin-producing cluster from the embryonic cell into a streptozotocin-treated NOD skid mice, it was demonstrated that hyperglycemia could be reversed for 3 weeks, okay? But, however, the long-term control of hyperglycemia failed. It became apparent that insulin production is lost at the tetramer formation move forward in the current study, okay? So, beta cell-like development in vivo after transplantation on human embryonic-derived pancreatic cell preparation, tetramer formation was observed at least in 50% of graft. That's huge. It's not minimal effect, okay? So you have a huge impact of tetramer formation, meaning it is essential that the encapsulation method does not link cell outside the capsule, okay, to succeed in this area, okay? So I will give you another example that sometimes scientifics like us are enthusiastic about our work, okay? I was in 1990 at the Institut Cochin Genetic Molecular in Paris with my friend, colleague Axel Kahn, and we do something that was extraordinary there. We are the first group to induce the hepatocyte to secrete insulin in a response to high-level glucose and to be shut down on hypoglycemia. How the way we do that? We simply take a promoter, the L-pyruvate kinase promoter. It was a promoter that when your glycemia is high, is induced by glucose, it simply switch on. And when the glycemia is low, the glucagon inhibit the promoter. So the logic was simply to bind the insulin to this promoter, put that on the liver cell, target the liver cell, and effectively, by spectrotomic and transgenic mice, we show perfectly that we're secreting insulin in the response to glucose. And it was a fantastic model we patented, but we face a wall. And the wall was simply this one. When you check here the cat activity of the promoter of the L-prerogat promoter gene, you see that when the glucose induces this promoter, it takes like 72 hours before or 48 hours before it touch the high level of activity. If you want a treatment for type 1 or type 2 diabetes that need insulin, you need to have an action that is minute to minute. It was too slow. And we accept that effectively, at the stage that we are in science today, we will not able to modify the transcriptional machinery that to express this promoter in higher level and more rapidly. So we move on, and we accept, but we prove that we can make something there, but we prove also that it was impossible to treat diabetes with this approach. We can secrete other protein than insulin, but not necessarily insulin because it was too slow. Now we have another problem in science today. We love this animal model, okay? But in reality, all this animal model study on beta cell formation came with what I call co-founding effect. And the co-founding effects are not low. Take an example, the streptozotocin. Streptozotocin by himself, a molecule that we love to use sometimes to induce diabetes in pancreas, induce beta cell proliferation by himself, okay? He also, streptozotocin induce the expression of the transcriptional factor NG3, implicate on beta cell formation just by himself. Streptozotocin is an alkaline property that can lead to DNA fragmentation and cause cell proliferation. And streptozotocin induce tumor in pancreas, in vivo, etc. So every time I use streptozotocin, I cannot use that because I said if I use my cocktail to induce beta cell formation, I have all this co-founding effect that is present. I will never be sure that my treatment works simply because I put in a system that will tell me what I want to see. Now take the NOD mice. They develop thyroid estrogen syndrome. They increase the activity of EL1 and inflammation, okay? That is also a co-founding effect that promotes cell proliferation. I put myself in a situation to win when I came with my treatment. Also, the skate mice rat developed spontaneous intestinal neoplasia. The right two mice developed spontaneous intestinal and mammalian tumor. So the immunodeficient mouse model, spontaneous cancer, create a co-founding variable when it will be unable to untangle the effect of molecular cocktail parser from the natural spontaneous cell proliferation effect of the model of your treatment you want to do. Now you have another problem. The genetic modified animal, transgenic or knockout, do not represent a good model to study complex disease such as diabetes. And more than that, the conditional gene distribution experiment using the Krelux recombinase was demonstrated to be toxic to the cell by damaging the genomic DNA as a result of the recombinase activity Krelux. Okay. So based on that, an example, we decide, something that we'll talk in my symposium, we decide to never use the model that I showed you before for the co-founding effect. We use a wild-type mice, okay? And now we came with this approach I call beta cell formation in vitro to cellular networking, integration, and processing in a wild-type adult mice, and insist on wild-type adult mice, okay? What we show effectively with our cocktail that I will discuss in my symposium, we're able to cure a mice that received partial prostatectomy for more than a year without any side effects, okay? And they are cured. But I'm using wild-type mice here, okay? But I insist on one thing. I insist that when you clear a mouse model, you stay a mouse, okay? Missing between mice and humans, majority of times, sometimes is the large animal model, longitudinal study need, like dog, pig, or monkey. The mice is the proof of concept. But in reality, after that, if you want to translate that to human, you need the large animal model that more close to the level of metabolism that you will find in human than a mice. And the reproducibility is a problem in science also, a huge problem today, because the study made by recapitulation of novel cell VIACID protocol to generate embryonic derived endoderm cell for implantation in epidermal fat of atomic mice, rat, with the tetracyc encapsulation device. And unfortunately, they were not able to reproduce the beta cell in vivo on sufficient beta cell production. Okay, they not reproduce the other company. Okay, what they do later, they report a success in deriving beta cell after implantation on a new rat with different human embryonic cell line implant the seed kidney cycle and modify the culture protocol, okay? It demonstrates the importance of the protocol, okay? The site of implantation and the selection of human embryonic cell line, okay? But in regard of that, still, all this study was based on a nude rodent that absolutely don't mimic the variability that we are human. We're not inbred mice, us, okay? And we face another problem. The cancer research error was go directly from mice to, and a good example in the past was this, what's happening in the cancer field. In a hematopoietic stem cell transplantation, the most important milestone are those relevant for blood cancer, still today. They have a definitively an impact in cancer treatment for blood, okay? And what you see is that the early clinical transplantation effort in the late 1950s and early 1960s failed because this procedure was based on the work in inbred mice, which do not require histocompatibility matching, okay? After testing with a dog model, they realized the graft donor problem. And at this moment, these failures have occurred mainly because the clinical applications were undertaken too soon, most of them even before the minimum basic knowledge required to bridge the gap between mice and patient had been obtained. Beta cell-like encapsulation at the periphery. Now imagine that your strategy works. You take your beta cell light, you put that on the capsule, and you put that on the periphery. Following the secretion of insulin into the portal vein system, 60% is subsequently removed by the liver. And hence the importance that the insulin pass by the liver first. Liver is an important organ. And this is important because after you don't have what we call the hyperinsulinemia in the periphery. Like people mentioned here, the problem of hyperinsulinemia, okay? And the capsule under your kidney create an hyperinsulinemia as insulin injection. So you will not take off the problem of hyperinsulinemia here. Advantage of beta cell-like encapsulation will do not induce hypoglycemia and longer need for insulin injection. Of course, that is an advantage. Definitively an advantage, so you go there, okay? So, the major argumentation for stem cell replacement and islet transplantation is to provide an unlimited source of beta cell light for transplantation. Can protocol described in the literature to differentiate beta cell light be replicated at appropriate scale for production still stay a huge hurdle today. Nobody know how to scale up that, okay? Can the encapsulation method eliminate the ultra-pure alginate from algae and bacteria that still contain microbe-associated molecular patterns such like liposaccharide, lipocoid, and they are highly immunogenic? It can enhance also graft failure. I want to remember that colonized isolation has still never succeeded to have a preparation without contamination, okay? This method dates from 1960, okay? Paul Lacey in St. Louis, Missouri. Longitudinal studies are obviously necessary in this area. How long will this cell function in vivo? We perfectly know that stem cell device continually divide and lose their phenotype, okay? So the old question that we need to ask about this. Will patients need to be retransplanted with new beta cell-like periodically if the graft cell become dysfunctional or die, okay? It's another question that the field have to ask at this moment. Self-congratulation in scientific culture, okay? Simplistic view of the marketing of stem cell today in the field, okay? You have headline commonly feature the work like breakthrough, first, hope, cure, stem cell pluripotency. You have also vulgarization of data present in scientific meeting and news. You have a high-impact peak review journal acting more as a marketing agency, etc., etc. It doesn't help the field of science, okay, as us, like a scientific, from the perception of the public, okay? And the clinical hypotheses are more speculative, the therapeutic mechanisms are incompletely defined, and some instances of the preclinical evidence is highly contentious. And that was a study making of who optimist, pessimist, or neutral in different countries like Canada, US, or UK, okay, and their percentage. So you have the perception of the population that effectively doesn't understand how science works in our laboratory, okay? So the field is gradually progressing, but the therapeutic rhetoric must be tempered and to reflect the current clinical and research reality in this area. Fundamental research, what I call basic science, are simply wiped out of the field by what I call translated researcher. We still need more basic science, art biology, based on this area. And we need more diverse scientific culture in the field of stem cell rather than a concentration of the same approach with multiple recipes to form beta cells that follow the same reductionist linear concept of gene expression observation. Science advanced by its diversity and variety of alternative approach, not by a repetitive concentration of effort. Okay. We definitively able, when you look at the biological system, to have so much different angle you can pick up. But when you go to conference, you see systematically the same angle. Okay. And we all want stem cell or alternative approach to succeed for our family, friend, or subject that deal with the disease every day. My presentation is not something against the stem cell. I just said the research have to continue in this field, but with optimism and realistic approach. And it will take probably more years to come to success than what is said in the news or publication at this moment. And no matter where the success comes from, we definitively need it for type 1 and type 2 diabetes. Okay, I'm stopping here. Thank you for your attention. It was just a general commentary. I can understand if you don't know. Yes. Yep.
Thank you. I'm trying to make sure I say this right. Doiron. Doiron?
Yes.
Dr. Doiron. Okay. Do we have any questions for Dr. Doiron? There we go, man. I said it perfect last night. I couldn't get it.
Well, I'm very happy. I really don't want to make question. You know that I am the founder of Beta-O2 Technologies, which is really a means for encapsulation of whatever cells. And we are stuck because whatever you say is true. We have the machines, but the cells are not responding to our desires. But I mean, what is very important, what you have said and what we all feel is that we have the concept that we all need very quick success. And this is because we are not doing actually science, but social science. And it's important that we come back and be human and really go to whatever it is science, which is the research for truth. But we are driven by ego trip everywhere, and this is, I'm telling about myself. We need this ego trip, and which is good, but it has to be also supported by a lot of efforts and a lot of work in the laboratory to achieve something. But we are all living in a materialistic world. And materialistic world is driven by money and economy. So I think we have a lot of, the physicians have a lot of power because we also can influence it, not be only influenced by economy. That's what I saw. So I think we need to understand how much power we have because the pharmaceuticals and whatever is giving the money needs us, but we don't understand it because we always run after them. And this is my personal feeling. Okay, I think we need maybe meetings like that to form small things, not the ADA, not the ESD, to make some relation between people and to have sometimes to understand what people say and to make some good research commentary, not just listen to something that we have listened for 20 years and feel that we are well appreciated because we come and we are invited to give a lecture.
Yes.
So thank you for your...
Thank you. you have a very nice presentation actually. So these stem cells actually they have been used experimentally in the animal models. So and then you have said already that they are ability to produce insulin. They can be converted into insulin producing cells and indefinitely, because this is their characteristics. They are unspecialized cells. They can be directed to specialized cell or insulin producing cells. Now
I will not say indefinitely, because they differentiate and they will lose their phenotype, okay?
Yeah, but the only difference is because in animal models, they stop producing insulin just after 2.5 years or 3 years maximum. So they convert it into the cancerous cells. But now there is a study done by some scientists in the Yale University. It is in 2013, I think. And they have discovered a marker which is going to tell us that which are the stem cells which are going to be converted into the cancerous cells and which are going to be converted into insulin producing cells. So now we have a technology. So this problem has been solved.
I'm totally not agree with you that the marker are be solved.
Yeah, they have discovered. In Yale University, I can give you the
maybe I'm not aware about this study.
Yeah, so this
but
is
all the thing I know that the last summit, worldwide summit in Boston in 2016, 2 months ago, I'll tell you that marker was still a problem.
Yeah, but they said
maybe a new study came, but I don't know this
now
study.
they have told categorically that we know that which are the cells which are going to be cancerous and which are going to be insulin producing cells. So when we have this technique, so we know that these are the good cells and these will not be converted into cancerous cells in future. So we can have a problem. It's solved almost. So
I will not say solve almost, because I don't know your study. So we can discuss a long time, but I don't know what you're talking about. So in this case, I will just say what I see for the last update that I was in a meeting 1 month ago in Boston, in the Stem Cell International meeting, when everybody was there, okay? Via SIT, everything like that. I see that the marker is still a problem. But maybe on your study find marker that I don't know, but the marker is still a problem in stem cell in general. The marker by definition only for hematopoietic stem cells still a problem, still today. So I know that the marker, because the marker came from this molecular fallacy logic, that when you have something that pop up, that give you the future of the cell, something like that, sometimes we miss our point, okay? But I can tell you that for me the marker is still a problem in stem cell.
But it was a Yale University study. I can give you a reference to more.
Other question? Okay, thank you very much. I think we pass now for the break, for the lunch break. Okay.